First thing I'd like to do in this video is very briefly go over what we covered in our last class, specifically walking through this big clunky diagram about our adaptive immune system. What I have for you is part of the diagram, so I want to point out first that all of this begins with a lymphoid stem cell, right, ultimately replicating and differentiating in the bone marrow. Okay, remember that the bone marrow is in our bones, and so these T cells, of course, cellular immunity, don't finish their development within the bone marrow. Instead, they are dumped into these sinusoid capillaries and are washed via the blood, ultimately into the thymus. Right? It's in the thymus that, as we see up here, that these T cells are going to be trained and they are going to differentiate. Right? So these T cells, by the time they get to the thymus, have both CD4 and CD8 co-receptors, right? And they also have T cell receptors. So at this point, they could still be any kind of T cell. Okay, so after they are trained in the thymus, right, we can go back down here, they are going to be dumped into the lymphatic vessels, or into the blood, and ultimately into the lymphatic vessels. And so, they ultimately get milked along these vessels, and they are going to populate in one lymph node, then the next, and the next, as they continue their journey. Remember that this is a lymph node. All of these little blue and purple cells in here are representing the T and the B cells. In this case, we are talking about the T cells. And so this is where a T cell, right, which is naive, it has either a CD4 or a CD8 co-receptor, so it is differentiated at this point, but it is still naive. That is, it hasn't ever been exposed to something for which it has been trained to recognize. Okay, so again, T cells are hanging out in these lymph nodes. In the event of a breach of the primary surface barriers of the body, right? So at the skin, right, or within the respiratory tree, within the digestive system, right? Any time that some kind of a pathogen crosses over one of these surface barriers, it is met, hopefully immediately, by an antigen-presenting cell, and so that's where we come in up here. This is the pathogen. Right? It's immediately phagocytosed or phagocytized by an antigen-presenting cell. So something like a natural killer cell, or even a B cell, a dendritic cell. So one of these cells that has a role specifically in engulfing these pathogens. As we know, after phagocytosis, those pathogens are then digested, and a piece of that digested pathogen is then presented on an MHC complex on the surface of the antigen-presenting cell. Now keep in mind that while all of this phagocytosis and digestion and presentation, all that kind of stuff, is happening as the cut, or after the cut happens, and these APCs with their internal pathogens are being pushed along, milked along in these lymphatic vessels, and then they go into a lymph node, right? And keep in mind that these lymph nodes are populated, of course, by these T and B cells. Okay, so that's where the APC would actually do the presenting to the naive T cells. And what we can see up here is kind of the story if a CD8 or a cytotoxic T cell were to recognize this pathogen first. Right? So what we can see is an MHC1 complex is going to present the antigen to a T cell receptor. This CD8 co-receptor is going to help with the binding process, so actually help the APC dock to the cytotoxic T cell. That's why the CD8 co-receptor determines what MHC conserved region is going to be recognized by the CD8 cell. So we have this recognition by the T cell receptor on the cytotoxic T cell membrane that, hey, this doesn't belong, right? This MHC complex does, but this does not. Okay? In order to actually fully activate the naive cytotoxic T cell, we also need something called co-stimulation. In this case, the APC is what is responsible for releasing interleukin 1 or IL-1, or a slew of other molecules, but in this case, interleukin 1 is released, and this co-stimulation fully activates the cytotoxic T cell. If this co-stimulation did not occur, the immune system, so these cytotoxic cells, would not recognize this particular pathogen in the future and wouldn't be able to eliminate it, right? And that is called anergy. All right, so once we get this co-stimulation as well as presentation, what's going to happen is a full activation of the cytotoxic T cell. Now we call it a mature T cell. What that's going to lead to is proliferation. Right, so this T cell would be hanging out here in the lymph node. It is now activated by presentation and co-stimulation. And so it's going to start replicating, undergoing mitosis after mitosis after mitosis. The reason why the doctor checks your lymph nodes, right, usually around here, to see if you're sick, is because every time you have a lot of mitotic activity, this lymph node is going to swell. Right? And as it swells, you can actually feel it through the skin. Again, that just means that these T cells have been activated and they are actively fighting something in your body. Okay, so once we have lots of these baby cytotoxic T cells, they are released into the blood and ultimately into the lymph, so the lymph fluid. So they are going to circulate all around the body, looking for whatever bears this particular structure. Okay. Whenever these cytotoxic T cells find one of these pathogens, they are going to release granzymes, right? So those toxic molecules to stimulate the pathogen to kill itself. Okay, so that is CD8 cells, right? CD4 cells, or the T helper cells, follow largely the same pattern, the same life story, right? So they are made in the bone marrow. They are trained down here in the thymus. After the training and further differentiation in the thymus, they are going to migrate out to the lymphoid tissues, again, to lymph nodes, to the balt, to the galt, to the tonsils, wherever. Again, just waiting in these tissues until they see something that doesn't belong. Here again, we have an antigen presenting cell that is literally presenting the antigen to the T cell receptor. Again, this docking is facilitated by CD4, and that co-receptor making this a T helper cell. Okay, once this presentation as well as co-stimulation by interleukin 2 occurs, we get again activation, it's now mature, and proliferation. So we make a ton of this particular T helper cell that recognizes this particular antigen. Okay, so these T helper cells are really the most powerful cells in all of the immune system because they enhance the entire immune response. Right? We have all of these other cells that have their own jobs and they actually go forth and kill things, but these T helper cells are needed to activate more of those cells in the innate immune system, so natural killer cells, macrophages, and others, and they of course activate the B cells. Right? So this here is a B cell which has already recognized an antigen. It has broken it down and presented it upon its own MHC2 complex. Okay, and so these T helper cells would then activate B cells, and only then would the B cells be able to make antibodies. Okay, so that, again, is a very brief overview of everything that we talked about the other day in class regarding the cellular division of adaptive immune system.
Now, before I continue with some new information and complete this big diagram that I've been talking about, let's do a couple review questions. Which is false about the natural killer cells? Well, do they recognize self versus non-self? By examining the conserved regions of MHCs. This is actually true. We know that the natural killer cells look at the MHCs to determine if cells are our own. And so if a cell is foreign, it will not have our MHCs. the same pattern of our own MHCs, and so the natural killer cells will phagocytose them. And so that leads us to B. Their primary role is to phagocytose abnormal cells, including cells like cancer cells. Okay. Are they antigen-presenting cells? Yes, they are. So not only do natural killer cells recognize that cells are cancerous by looking at their abnormally shaped MHCs, but also they can see that a virus has infected cells, and so we can actually phagocytose virus-infected cells, chop them up inside the natural killer cells, and present them to our adaptive immune system cells, and therefore launch a systemic immunological response. And we can also remember this for next time. Okay, so A, B, and C are in fact true. D, respond faster and stronger to a pathogen the second time it invades your body. This is false. Natural killer cells, again, are part of our innate defenses, not our adaptive defenses. And so the answer here would be D. Next question. Blank is the absence of an immune response to a particular pathogen. All right, so that's our first clue, particular pathogen, not generic pathogen, any pathogen that comes in. So this is something to do with our adaptive immune system. Specifically, this is often due to the lack of simultaneous antigen presentation. Right, so we know that this here is again part of our adaptive immune system. So essentially an APC presents a piece of a pathogen to a T cell or to a B cell, and the simultaneous co-stimulation of the T cells. Okay, so this is here, this is what we should go after, and no, really, this is dangerous, right? So both of these messages at the same time. And so if we don't have both at the same time, what is produced? Is it A, immunosuppression? Right, so actually actively suppressing the immune system. This is not the appropriate term. B, immunocompromised. This is a side effect of a lot of different conditions, such as if you are undergoing chemotherapy, if you have had an organ transplant and are taking medication so that you don't, quote unquote, reject that foreign organ, or in general, if you are otherwise sick and unable to produce this kind of response, elderly, super young, etc. And so no, that is not the case. Anergy is the correct term here. Okay, so anergy means that you don't produce a response to a particular pathogen because you don't get the simultaneous presentation and co-stimulation. An autoimmune is essentially an overactive immune system, so you cannot recognize self versus non-self, and so you start to have an immune response against self cells. Next question. T cell receptors of CD4 cells recognize pathogens only if they're presented on these protein complexes. So the CD4 are the helper T cells, and so I can tell you that the answer here is class 2 MHC, and this one really is just some rote memorization. Final question. These cells release histamine, so essentially the process called degranulation, at the site of infection, triggering the inflammatory process. Okay, so this is at the site of infection. It is not part of our adaptive immune system. It is directly triggering inflammation, right? So both the site of infection as well as the inflammatory process both tell us that this has to be some kind of cell within our innate immune system. And so it's not a T helper cell, it's not a cytotoxic T cell, it's not a B cell, and so therefore it kind of has to be a mast cell.
All right, now we have zoomed out. All right, so what I want to show you is, the same stem cell. Here are the two pathways of the T helper cells and the cytotoxic T cells. Now I want to focus on this humoral immunity, so the B cells, right, again made in kind of the same place, and then we ultimately get a slightly different pathway. This component down here, we did not get to, and so I'm going to elaborate on that in this video. Okay, so just to review what we have talked about so far, here we have differentiation of daughter cells of these lymphoid stem cells within the bone marrow. We get the growing up and the training, and ultimately killing or remodeling of these cells if they aren't quite up to the task. Any successful B cells are then, again, sent to the lymphatic tissues. Right? So they spend their entire developmental life within the bone marrow. They don't go to the thymus or anywhere else. And when they're finally ready, they're going to migrate to the lymphoid tissues. Okay, so again, this is an image that we've seen before. This lymphoid stem cell can become a B cell, or of course over here, a T cell. Okay. These cells are going to migrate again to the lymphatic tissues, such as a lymph node here, the tonsils, the malt, the galt, anything else. Right. The next couple animations here, you're going to see some text. This is specifically taken from your notes, right? So it's elaborating here that B cells recognize specifically extracellular pathogens, parasites that are either free or preferably actually presented by an antigen-presenting cell on a class 2 MHC. Okay, when these B cells actually recognize the antigen, which we can see up here, they are going to follow pretty much the same pathway as any other antigen-presenting cell. Right? So they are going to phagocytose this and present it on the MHC2 complex. Right? I have brought this image up as well. This is also in your notes, so you can follow along with that as well. Here we have the antigen. It is recognized by a very specific type of antibody on the B cells. Only B lymphocytes can express antibodies. Okay, once this recognition and binding has occurred, we get phagocytosis and ultimately presentation on an MHC. Okay? This B cell is not yet fully activated. In order to be activated, we need our friends, the T helper cells. Right? And remember that these T helper cells cannot actually be mature. They can't actually go forth and activate the rest of the immune system if they themselves haven't been activated and co-stimulated. Okay, so all that's happened before this particular moment in time. Okay, the T helper cells, if they have been activated, co-stimulate these B cells, and that is the event that actually triggers the production of more B cells. Okay, this process is called clonal expansion. Okay, clonal expansion essentially creates thousands more of these particular B cells. So the B cells that have this particular antibody, not one of the other millions of antibody structures, but this particular antibody. Okay? And these clones don't all have exactly the same role. Some of these clones will ultimately become plasma cells, and these are the ones that are making antibodies right now. As I said, these are the ones that are secreting about 2,000 antibodies per second per cell. We have lots of RER, rough endoplasmic reticulum, to continually crank out all of these antibodies. Okay, we'll get into what they do in just a moment. But the other option for these clones, again, of the activated B cell, are memory cells. Okay, so that was a summary of humoral immunity. Right, so cellular immunity deals with T cells, humoral immunity deals with the humors or the fluids of the body, and so the B cells and their production of antibodies are said to be the humoral adaptive immunity. Okay? Remember that there are millions of different types of these B cells, but only the ones that have previously been activated, that is, that they or their ancestor cells have actually seen and detected the pathogen that they themselves are able to recognize, and they have been co-stimulated, activated, and finally undergo clonal expansion. Okay, essentially, the benefit to this is that we can produce thousands of these cells with the same exact antibodies all at the same time, so we can really hammer that particular pathogen whenever we are exposed to it. Okay, again, for the first exposure, for the current exposure, most of these clones become plasma cells. They're the ones that secrete antibodies for the next 4 to 5 days. Okay, so that's hopefully enough time to fight this infection. Some of these plasma cells keep on living, and they're going to populate the lymph nodes and other types of lymphatic tissues, ultimately keeping the levels of antibodies higher until after the infection is fully dealt with. Okay, so that's where we're going to go now, but just a little preview. These memory cells are the ones that provide immunological memory. That is, the adaptive immune system has to first be exposed to something, right? Making many of these B cell clones, and then and only then can we remember that particular pathogen so that we can fight it better in the future. Okay, so the plasma cells are capable of producing thousands of antibodies every minute. These antibodies have a general structure, and this might seem a little bit similar to the MHC in that there is a constant region and a variable region. Generally, antibodies are shaped in a T or a Y, and we'll see a little bit more about that here shortly. This variable region forms two different antigen binding sites that are identical to each other, but unique among all of the other antibodies. So we can see that this forms a very specific key to fit a particular lock, right? So a very specific binding to antigens. The reason that there are two is that one antibody can bind to two antigens at the same time and actually clump them together, right? So this is agglutination, which we've seen before. There are also a couple different types of chains, right, protein chains, and actually I have a short video that I'm going to embed in this that you can watch as well that elaborates on the protein chains themselves. Okay. Really, one of the take-home messages here is that the constant region, so down here, the one that is the same for every class of antibodies, determines the identity and the behavior of that particular type of antibody. Okay. Again, I'm going to embed a video within this PowerPoint that's going to walk you through the basic structure of antibodies and all 5 of the antibody classes, so immunoglobulin M, A, D, G, and E.
The immune system depends on special binding molecules called immunoglobulins. These are also known as antibodies. Immunoglobulins are often referred to in shorthand as simply Ig. Igs are Y-shaped molecules which connect on one end to an invading microbe, which is known as an antigen, and then they connect on the other end to bind with various white cells. These specialized antibody molecules come in different shapes and sizes, which provide a great deal of flexibility in matching and destroying targeted antigens. Antibody molecules are found floating in the plasma as well as on the surface of specialized B cells. Immunoglobulins are constructed from 2 types of amino acid chains, heavy chains and light chains. Ig molecules contain 2 identical strands of heavy chains and 2 identical strands of light chains. The side of the Ig that has 2 heavy chains forms a stable non-variable receptor point called an effector. This effector is part of the Ig molecule that binds with our own immune system cells. On the opposite end of the antibody are 2 antigen binding sites, each with a light chain and a heavy chain combination. This combination of light and heavy chains forms highly variable tips that can adapt to specifically match a wide range of antigens. Immunoglobulins come in 5 basic types: IgA, IgD, IgE, IgG, and IgM. Each type targets a specific type of antigen, which determines the wide chain structure of each type of Ig. IgD, IgE, and IgGs all use a single Y chain, whether they stand alone or as receptors connected to white cells. IgAs use a double Y chain, which is called a dimer. IgMs are unique in that they form either as a single Y chain when expressed on the surface of a B cell, but the standalone version of the IgM molecule forms a combination of 5 Y chains, which is known as a pentamer. Consequently, standalone IgMs form a very large antibody molecule. In the next video, we'll look more closely at the functions of each of these 5 Igs. Let's take a closer look at the 5 types of antibody immunoglobulins. There are 2 types of IgAs. These are not typically found in the blood, but in secretions of the gut, saliva, sweat, milk, respiratory tract, and urinary tract. IgAs are part of our innate immune system and fight pathogens such as bacteria, viruses, fungus, and parasitic worms. IgDs are primarily found on the surface membrane of B cells, but account for a very tiny portion of the free-floating antibodies found in the plasma. IgDs are the least well-known antibody. However, they are known to play a role in the activation of basophils and mast cells to attack invading microbes. IgEs respond to parasitic worms as well as allergens such as pollen. The effector side of the IgE is ideally matched to mast cells and basophils. And once the receptor side of the IgE comes into contact with one of these antigens, the mast cell and basophil will release histamine molecules, causing the inflammatory symptoms we know as an allergic reaction. The 4 types of IgGs account for 70 to 80% of the entire immunoglobulin pool in the body. IgGs target pathogenic organisms such as bacteria, viruses, and parasitic worms. They attach to the antigens on one end, while the opposite end binds with a phagocyte white cell, including macrophages, neutrophils, and natural killer cells, which absorb and destroy the antigen. IgG is the smallest of the Ig molecules, allowing it to easily migrate into the tissues of the body. Only 45% are found in the blood. Almost every man-made monoclonal antibody in clinical use today is in the IgG family. The early responders are IgMs. These function to eliminate pathogens early on in the attack before there are sufficient levels of IgGs. IgMs are known as a macroglobulin because of their large size. They are the largest of the immunoglobulins and make up 6 to 10% of the total Ig pool.
The previous videos just showed you and introduced you to what general antibodies look like, the different protein chains, as well as the different structures on them and how they actually work. You also saw that there are 5 different types of antibodies and that they are each found in different places. So I want to, at this point, summarize them and give you some take-home messages very briefly. Here, what you can see in this chart are the 5 different immunoglobulins or antibodies, M, A, D, G, and E. We can see off to the side here that each of these has a different shape. So IgA is a dimer, D, E, and G are monomers, and so these are more common, and then a pentamer with 5, and so these produce like super globby antibody-antigen complexes. Okay, so take-home messages, something that you might expect to complete some matching questions about on an exam. IgA is found in mucosal areas, right? So you can think about places like the oral cavity, the bronchial tree, the digestive system. That is where you would find IgA most abundantly. IgD is the most common receptor on B cells. Okay, so the ones that are actually existing on the B cells that first recognize that antigen even before the B cell has been activated, those would be ID or IgD. Most of our antibody-based immunity is IgG versus IgE. These are the ones that produce more of an allergic reaction. Okay, and remember that we talked about how we have two different types of T helper cells. One produces IgG, the second produces IgE, and having a balance between these two will produce an appropriate immune response, not allergies or immunodeficiencies. All right, so what exactly is the big deal about antibodies? Why do we spend so much time and resources into making antibodies and, of course, making vaccines to trigger the production of antibodies, etc.? Right, so just a reminder, antibodies are secreted by plasma cells. Plasma cells only exist and are produced after clonal expansion of a particular type of B cell. Right? So whatever B cell has this particular antibody that recognizes this particular antigen is going to be activated, and therefore these antibodies recognize the same antigen that we saw up here. Okay, so what's the big deal? Why are antibodies so important? They do not kill anything. Right? So we're not making antibodies to go forth and just slash everything that looks like this particular antigen. No, they're not killing anything. They're merely promoting the destruction of this particular antigen. Okay, and so they do that in a couple different ways. First of all, as we see over here on the side, they neutralize the threat. Okay, if you've taken microbiology, you might know that one of the reasons why bacteria and such are so dangerous is because they're so sticky. Right? So they get into our bodies and they stick to our tissues, and then it's really hard to get them out once they stick to us. And so neutralization essentially creates a physical barrier between the very sticky bacteria and our body's tissues, and literally the antibodies just stick to their antigens on the surface of the bacteria and create a little buffer between the threat and our body. Okay. Secondly, we can promote phagocytosis, and we do that in a couple different ways. Some of these both of these we've already heard of. Opsonization is part of a process activated by complement. We'll talk about that again in just a moment, but this process again is linked to our innate immune defenses. Okay, but the production of antibodies really ramps this process up. Essentially, opsonization is coating the bacteria kind of like neutralization does, but in this case, the function of this coating is essentially to flag down any phagocytes. Okay, so each one of these little antibodies is essentially saying, hey, like macrophages, natural killer cells coming over here, this doesn't belong, we need to get rid of it right now. And so the rest of this image here is essentially part of a phagocyte engulfing this bacteria, which of course is covered with antibodies. Again, antibodies are not killing it, but they are promoting phagocytosis, which is indeed killing these things. And the second way of promoting phagocytosis is agglutination, and we've seen this before when we talked about the red blood cells. What we can see in this image is one of those IgM antibodies, right, with lots of, or not lots, with 5 antibodies in this pentamer construction. And so what we can see is multiple mismatched red blood cell types are bound to this one single antibody, creating this big, gloopy, clumpy, clump. And so these don't circulate in the blood freely. They are going to precipitate out of our circulation, and once they do that, it's very easy for a phagocyte to come along and engulf these and break them down and get them out of our blood. Okay, so again, both opsonization and agglutination are promoting phagocytosis, and phagocytosis will actually kill, right, but not the antibody itself. Okay, and the third and final action that we're going to talk about of antibodies is kind of ramping up complement. Again, you've heard about this before when we talked about the innate immune system. What's going to happen is a series of proteins within the blood is activated, and that ultimately leads to more inflammation and more destruction of pathogens. Specifically, where these antibodies come in is that they're going to bind to the surface of these pathogens, again, just like always, and essentially that calls over these complement proteins to essentially invade the pathogenic cell and cause cell lysis, right? So actually like bursting of the cell, therefore killing it. Okay, so again, I say that the antibodies are not killing anything. Okay. They are merely neutralizing the threat, promoting its destruction by macrophages, and inducing complement proteins to kill the cells from the inside out. This is just another way of looking at the functions of antigens and antibodies. This is the pathogen, this is the antibody. When they join together, they do all of these different things, right? It activates complement, which leads to cell lysis or kind of exploding, so this is cell death. Also, complement enhances inflammation and phagocytosis. Antibodies can also inactivate the antigen itself by neutralizing it, agglutinating multiple together into big clumps, and of course that facilitates precipitation out of the solution. Okay. Immunological memory, right? So this is a function of only the adaptive immune system, not the innate immune system. Okay, what we can see in this diagram here is that we have lots of different types of B cells, each one recognizing something a little bit different, right? A purple something, a red something, a blue something. When a particular antigen is detected by the very specific antibodies, only those cells clone themselves, producing mostly plasma cells, but of course then we have these memory B cells. Okay, these memory B cells, and of course memory T cells on the other side, promotes immunological memory. And so the idea here is that the next time this same antigen is detected, we already have the activated cells here, and these cells can then go forth and clone themselves, making lots of plasma cells, and immediately produce antibodies. You don't have to go through the whole process of phagocytosing and presenting and being activated by T helper cells and ultimately undergoing mitosis and so on and so forth. All of that takes time, and therefore the primary immunological response is weaker than the secondary. Right? And that is going to be shown to you here in this graph. So this is time. This is when your initial exposure would be to whatever this pathogen is. This here would be the secondary exposure. And what the red line is showing you is the concentration of the antibody. So how many of these are actually inactivating the pathogens invading your body. Okay, so what we can see here is somewhat of a shallow slope, right? It takes a couple days to really get enough antibodies to fight this particular exposure. Okay, after a few days, we see that the antibodies go back down if, of course, the infection has been reduced or eliminated. After this primary exposure, we can see that we never completely get rid of those antibodies. We still have these memory cells which are still capable of producing antibodies. So we always have a little bit of a presence of these antibodies in our system. And so the very next time that you're exposed, the secondary exposure, what we see is absolutely no delay. And we also see a really steep slope, so we're producing lots more antibodies a lot more quickly. You can see that this is the maximum immune response after your first exposure, and this is the maximum response of your second exposure. And so this is certainly beneficial to us to remember pathogens and have the ability to fight them right away. You are about to watch a clip from a crash course video on this secondary immune response and how we actually acquire this immunological memory.
The humoral response allows your body to achieve immunity by encountering pathogens either randomly or on purpose. Active humoral immunity is what we were just talking about. It's when B cells bump into antigens and start cranking out antibodies. This can occur naturally, like when you catch the flu or get chicken pox or pick up some nasty bacterial infection, or it can happen artificially, particularly through vaccination. Most vaccines are made of a dead or extremely weakened pathogen, and they work on the premise that because a secondary immune response is more intense than a primary response, by introducing a pathogen into your body, you're priming it to fight hard and fast should that antigen show up again. In the case of typically non-fatal infections like the common flu, this immunity should at least spare you from some of the most severe symptoms. But in the case of more serious diseases like polio, smallpox, measles, and whooping cough, vaccinations can be truly life-saving. Now, some antigens, like those for mumps or measles, don't really change much over time, so a few immunizations will leave you set for life. But others, like influenza, are constantly evolving and changing their surface antigens, so immunity to last year's flu probably doesn't work against this year's flu. Still, acquired immunity doesn't have to be active. Babies, for example, naturally obtain passive humoral immunity while still in the womb. They receive ready-made antibodies from their mothers through the placenta and later on through breast milk. And that works pretty well for a few months, but the protection is temporary because passively obtained antibodies don't live long in their new body, and they can't produce effector cells or memory cells, so a baby's own system won't remember an antigen if it gets infected again. You can also acquire this kind of temporary passive immunity artificially by receiving exogenous antibodies from the plasma of an immune donor. This is what recently saved some doctors and nurses who had contracted the Ebola virus from infected patients. A serum was made from the blood plasma of other medical workers who had been infected and survived. The antibodies helped defend the patients from the virus before their own active immunity could identify that particular antigen and start creating their own antibodies. It's not the same as a vaccine, which immediately engages your B cells, but it can buy a patient some crucial, life-saving time against an infection that would otherwise quickly kill. But
The methods of acquiring antibodies into your body, that is, the methods of acquiring humoral immunity, can be summarized into active versus passive. We have been talking about naturally acquired immunity. Of course, actual contact with the pathogen is what leads us to become sensitive and actually produce these antibodies that recognize the pathogens. What the video just told you, so what the crash course just summarized, was artificially acquired. So that is, your own immune system doing the active work, right? So actually replicating B cells. B cells themselves are producing antibodies, against, whatever the pathogens are. Of course, this is artificially acquired because this is simulated by human beings, not by just a chance exposure to a pathogen. So that's active. We also have passive methods of acquiring humoral immunity. Naturally acquired, so these are methods that have evolved into our species, things like antibodies passing from mom to placenta, or mom to fetus via the placenta, as well as via breast milk. Again, completely natural process, has evolved in our species, but it doesn't involve an individual making their own antibodies. And finally, artificially acquired, there are certain circumstances where your immune system is so very compromised that you can actually receive injections of concentrated immunoglobulins, so concentrated antibodies from other individuals.
Now, of the 4 different methods of acquiring immunity, or essentially immunological memory, I want to spend just a little bit more time focusing on vaccines. So this passive method of naturally acquiring immunity. So of course we've talked at this point about how breathing particles in and whatnot can get us sick, and of course our immune system responds to those particles the first time, but then the second time we are exposed to that same substance, we have immunological memory. The secondary response is stronger and it is faster, and so that is a completely natural way that our acquired immunity works to protect us, both now as well as in the future. And so I want to show you guys another clip giving you just a little bit more detail about how vaccines themselves work. And so of course the take-home message here is that what is actually injected into your tissues are just pieces of the pathogen, so just the epitopes or an inactivated or dead virion or bacteria, etc., not the live substance. And so those pieces are actually enough for the antigen presenting cells to actually show the T cells and the B cells and ultimately produce a primary response, so that the next time you are exposed to that same thing, you produce the stronger, faster secondary response. And I also wanted to point out that vaccines aren't just those pieces. This is something that the video itself doesn't go into, so I just wanted to add this. There are also adjuvants, so essentially extra substances which enhance the immune response, so that we don't need to inject quite as much or quite as often. And finally, there's also the fluid in which these pieces, these components reside, and that is some type of oil. Over the course of history, this oil has been cottonseed oil, it has been peanut oil, which has its own implications, but just to be completely clear, vaccines have these different substances in them, but the active component is the epitope itself, right? So the pieces of the virus or the bacteria. And so here is another short video, which is in a little bit more detail about developing the flu vaccine in particular.
Vaccines and the immune response. How vaccines work. Influenza vaccines are able to trigger an immune response by mimicking viral infection. They are usually manufactured using inactivated or killed virus particles taken from various circulating influenza strains. Inactive fractioned viral components contain the subvirion particles hemagglutinin, HA, and neuraminidase, NA. In Canada, influenza vaccines are administered via intramuscular injection. These fractioned particles containing the foreign antigens hemagglutinin and neuraminidase are released from the vaccine into the bloodstream. There, they are met with an immune response mediated by various immune cells, including macrophages, T lymphocytes or T cells, and B lymphocytes or B cells. A major mechanism of action involves macrophage phagocytosis of hemagglutinin, breaking it into smaller components. After ingestion, macrophages display the hemagglutinin antigen on their surface in combination with a specific receptor known as the major histocompatibility complex, or MHC. T cells are now able to recognize and bind foreign antigens that are associated with the MHC. Upon binding to the MHC receptor, T cells become activated and proliferate into either cytotoxic T cells, regulatory suppressor T cells, or helper T cells. Activated helper T cells express hemagglutinin receptors specific to the vaccine strand on their surface and play a major role in antibody generation and memory B cell activation. Unlike T cells, B cells are able to ingest hemagglutinin independent of the MHC. Once internalized, B cells process the hemagglutinin antigen and present it on their surface in combination with an MHC. When activated helper T cells interact with activated B cells expressing antigen MHC receptors, they begin secreting lymphokines, which have several effects. Lymphokines trigger activated B cell proliferation, which leads to either their differentiation into memory B cells or into plasma cells. Plasma cells produce hemagglutinin antibodies specific to the strain of influenza contained in the vaccine. Memory B cells aid in future immune response when exposed to an active influenza virus. When an infected host sneezes towards an uninfected person, the nasopharynx is exposed to aerosol droplets containing whole, live influenza virus. Once inhaled, the influenza virus attempts viral colonization of nasopharyngeal epithelial cells. Assuming that the vaccine strain matches that of the live virus, hemagglutinin antibodies block viral attachment of live influenza virus to host epithelial cells, and overall disease is either avoided or diminished in severity.
Now that we know a little bit more about how vaccines work and how they produce this immunological memory even when individuals aren't exposed to the real virulent strain of these pathogens in the first place, I do want to talk a little bit about what vaccines have done for the human species and for individual populations, and talk a little bit about herd immunity. This graphic was printed a few years ago in Scientific American, and what it shows us is the prevalence of different diseases over time. And so here we can see 1600 and going all the way up to, 1800s and going all the way up to the 2000s here, and this was published in 2017. The red dots here are showing us how many cases there were in any particular year. So obviously these very large bubbles indicate that there were a lot of cases in this particular time frame. Smaller dots indicate that there were hardly any cases that particular year. There are a series of diseases, right? So polio, pertussis, diphtheria, measles, hepatitis A and B, mumps, rubella. And so the interesting thing that I want to point out here is the vaccine came out around in this time here. So wherever we see the black dot, that indicates that a vaccine was developed and began to be administered to the general populace. What we can see in this graphic is that before the vaccine, there were many cases. This was a rampant disease, measles was, and then all of a sudden, vaccine came along, and then it was virtually wiped out. And the next video clip that I have for you, which is actually the last one of this lesson today, is going to show you measles and the vaccine and why there's a little blip and it has made a reappearance here. So stay tuned for that. But the overwhelming consensus of this data is that as soon as there is a vaccine available, it starts to be distributed on a wide scale, the diseases themselves virtually disappear, or they disappear entirely from a population. And so as long as a certain percentage of the population is receiving this immunity, whether actively or actively naturally or passively via a vaccine, this virtually eliminates the diseases from the population. And so this leads me to point out this concept of herd immunity. And again, in the last video clip, in a few moments here, he's going to talk about herd immunity, but before he does, I want to point out this graphic right here because I think it is really powerful. What we have are 6 different populations. This population up here is not vaccinated, so in this case, our population in November of 2020 does not have any vaccinated individuals to COVID-19. And so this would be us here versus increasing amounts of the population being vaccinated, so up to 95% right here. And so these blue dots here are unvaccinated. Right? And when these little blue dots turn black, that means that they are infected. And the red that you're seeing keep popping up on the screen is the transmission of the disease from one individual to a nearby individual who is not vaccinated. Now, of course, the disease in theory cannot be passed to a vaccinated individual, or at least it won't be passed, or at least the vaccinated individual will not experience the same severity of the symptoms as an unvaccinated person would. Again, let me point out what exactly is happening here. So this person becomes infected, they are unvaccinated, they interact with another individual because they are closer than 6 feet, they touch each other, they hug them, whatever. And so this next unvaccinated individual is going to become sick, but all of the people around that person happen to be vaccinated. And so the transmission to other people is going to be very slow because most of those people already have antibodies built up against this particular disease. But the more unvaccinated individuals that you have that are near each other, of course, the higher the likelihood is that the disease will be transmitted to them. And so we can see that as the percent of the population that's vaccinated goes down, we very quickly get this explosion in the disease. And essentially we can see that the transmission is increasing exponentially because each one of these individuals can pass the disease to as many people as they encounter, right? It's not just one-to-one here, it's one to as many people as get close enough to breathe each other's essentially lung droplets that are filled with these virus particles. Okay. And of course, 50% of the population, we can see virtually the entire population is going to be infected very rapidly. Of course, this is what we're dealing with as of November 2020. None of the population is vaccinated yet because there is not a vaccine available to us, and so we all stand a chance of acquiring this virus, this disease, because we don't already have the antibodies built up against it. Okay, so on another note, let us now transition into the final clip of this lesson together today. Another crash course video, this time about herd immunity and the measles and the role of vaccines in both eliminating the measles as well as measles making a comeback when vaccines were not utilized as much as they should be, of course, to create this threshold of what is called herd immunity.
Here in the US, a disease that was all but extinct has made a comeback. Talk about measles. It usually starts with flu-like symptoms and a raised rash on the face, followed by inflammation of the eyes, and then fevers as high as 40.5 degrees Celsius, that's 105 Fahrenheit, and in the very young, brain damage, hearing loss, and even death. This disease has been so rare in the US that it was pretty easy for scientists to figure out where the outbreak started. In December, a person carrying the measles virus visited Disneyland in California. Now, more than 100 people have been infected, and it's spread to 17 states. Now, yeah, 100 people might not sound like a lot, but keep in mind that that is twice the number of cases the US usually sees in an entire year, and that the outbreak is only 2 months old. Now, the Centers for Disease Control is concerned that the outbreak could gain a foothold unless more people are vaccinated. So, how does something like this happen? I'm Hank Green, and this is SciShow News. In the year 2000, scientists declared that measles had been officially eliminated from the United States. Elimination, in a medical sense, means that the disease is no longer endemic or regularly found in a certain region. But eliminating the disease in the US didn't change the fact that it was still prevalent in other countries, like the Philippines, where the current strain of measles seems to be from. And it remains a leading cause of death among young children worldwide. Though, thanks to vaccinations, worldwide measles deaths have decreased 78% in the last 10 years. Measles is a viral disease that's communicable through airborne droplets of water, just like the flu, and the viruses can linger on surfaces for a couple of hours. One infected person will infect, on average, 12 to 18 other people. That's one of the reasons why this outbreak has spread so rapidly. But of course, there is a vaccine for measles. It's been around for 50 years. It uses a small amount of live measles virus that's been weakened by a process called cell culture adaptation. This process basically genetically modifies the virus by allowing it to grow inside of some kind of medium that isn't human cells. In the case of the measles vaccine, we do this basically with like chicken embryos in a petri dish. After a while, the virus adapts to infecting chickens, and these adaptations make it less virulent to humans. While a natural measles virus reproduces thousands of times during an infection, a weakened virus can only reproduce about 20 times inside of a human host. That's just enough that it doesn't cause any symptoms, but it does stimulate an immune response in our bodies, which allows us to produce antibodies. Throughout the rest of our lives, those antibodies will be able to identify a measles virus so that our immune system can be awesome at destroying it. But the system isn't quite perfect. 3 to 5% of the population can still contract measles even if they're vaccinated. And yet, if a high enough percentage of the population is immunized, the virus can't spread. Instead of each infected person creating 12 or 18 new disease vectors, the average person creates less than 1 new disease vector, and thus the outbreak just doesn't happen. This, my fellow herd members, is called herd immunity. And it means that an entire community can be protected even if a few of those members aren't immune to the pathogen. In the case of the measles, 92 to 94% of a population has to be immunized in order for herd immunity to protect the whole group. So, how do we know all of this? Well, a widespread vaccination program for measles, mumps, and rubella, all airborne viral diseases, began in the US in 1963. And as the country's herd immunity increased, the incidence of measles went from 3 to 4 million cases a year in the 1950s down to just 37 cases in 2004. But in recent years, immunization rates have began declining, due in part to growing misconceptions that have erroneously linked these vaccines to autism and other disorders. As a result, in nearly 500 public schools at ground zero for the current outbreak in Southern California, the immunization rate has fallen below that 92% threshold, which has given the measles an opportunity to spread again. But people who've contracted measles from this latest outbreak are all over the country. More than half of them weren't vaccinated. Immunization rates in 8 other states are even lower than California's, with Colorado coming in last at around 81%. So this is probably not the last we've heard from measles. Our herd is being compromised. But how did we get here? How did we get to the point where people stopped using safe and effective protection against a disease and decided instead to take their chances? We'll be talking about the science of why we sometimes ignore science in a special infusion next week, so be sure to join us. In the meantime, thanks for watching SciShow News. If you want to help us share science with the world, you can become a supporting subscriber at subbable.com/scishow. And if you want to keep getting smarter with us, you can go to youtube.com/scishow and subscribe.